Or well, that was a pretty interesting experience. I've never had quite an introduction, quite so extensive, quite so laudatory. So thank you so much for that, David and Peter. And thank you to everybody for showing up. I thought perhaps it would just be me and the lab and Peter that would be here this afternoon. So much appreciation for you coming. I'm going to talk today about one of the projects that's really central to what we do in the lab. And maybe it will let me. There we go, got it. I'm going to talk about the portion of the work in the lab that focuses on neuroendocrine cancers. So neuroendocrine cancers represent a relatively small proportion, 2% of diagnosed cancers. They arise from neuroendocrine cells or endocrine cells in a variety of different tissues. These tend to be small proportions of the cells that are found in different organs throughout the body. The tumors tend to be very slow growing, and they're often very poorly recognized and poorly diagnosed. It's not uncommon for individuals who have a neuroendocrine tumor to take 5 to 7 years for actually getting the correct diagnosis of that disease. They may be misdiagnosed once or twice or even multiple times before the tumor is actually diagnosed. And that has a lot to do with a number of different features of this disease. One is that most of these tumors are hormone secreted. And so as a consequence, we see that they will have many of the side effects associated with overexpression or oversecretion of hormone. And that can mask the initial symptoms of the tumor so that they're not recognized easily. And that's what we have the mascot here of the North American and Canadian tumor societies. And it's based at the zebra, which is based on something that many of you might have heard or perhaps have said in teaching medical residents and students, which is when you hear hoofbeats, think horses and not zebras, because neuroendocrine tumors tend to be much less common than are the phenotypes and pathologies that are associated with hormone overexpression. So those are more likely diagnoses. So as a part of all of this, we see that neuroendocrine tumors can be really hard to identify, can be really hard to diagnose accurately. And they're also very slow growing. So we don't have really good models for working with these tumors and trying to establish cause and try to establish novel therapeutics. And as a consequence of that, we actually have limited possible interventions for many of these diseases and still fall back on surgery rather than potentially more exotic therapeutics that are currently available for other cancers. So, and apparently you all know the punchline, which is that my lab actually works on the RET receptor type. Everybody's told you that already this afternoon. My lab is interested in the RET receptor. RET has a number of features that are associated with neuroendocrine tumors. We're going to talk about it in relation to that disease as opposed to its other effects today. So RET is a receptor tyrosine kinase. It sits on the cell surface and transduces signals intracellularly. It's activated by a complex of proteins, including the neurotrophic factors of the GDNF, glial cell line-derived neurotrophic factor family, and a co-receptor that sits on the cell surface, which is a member of the GFR alpha family. When RET becomes activated by its normal ligands, these 3 molecules form complex. And because of the co-receptor's GPI linkage, it recruits it into special areas of membrane termed lipid rafts. These are cholesterol-rich regions of the membrane that are stuffed with all kinds of signaling. So they're very rich environments and domains for signaling downstream of a receptor to happen. When RET is normally activated by this kind of mechanism, you see that it has many roles in development in the normal body. So it's required for the normal development of the kidney and for the enteric nervous system. It also is important for maturation of peripheral and central nervous systems and for brain dopaminergic neurons, survival and maturation. But it also is involved in a number of endocrine cell types, including thyroid, adrenal, pituitary, parathyroid, and pancreas, and in the neuroendocrine cells in those tissues. There are a number of pathologies, as Peter alluded to, associated with mutations in the receptor. So if you lack the RET receptor congenitally, That can be associated with Hirschsprung's disease, a disease of improper innervation of gut, and with kidney agenesis, kidney abnormalities. But the activation, normal activation in tissues of the RET receptor has also been linked to other pathologies as well, including risks of Parkinson's disease, dementia, and other disorders, including depression. So there's a lot of variety of the kinds of disorders that might be linked to proper RET function. But in our hands, in our lab, we're really interested in RET and its role. Because RET is a potent oncogene. This is an example, an ancient example of work done by a PhD student at the time, Turan Bijwal, where he took NIH3T3 cells, or cells that had been transduced with an oncogenic form of RET, and injected them subcutaneously. And you can see really clearly that, in fact, the mutant form of RET is associated with rapid proliferation of these cells in the animal model. We know a lot about the kinds of things that RET is involved in cancer. It's got a number of different functions and different roles. So we know that the wild type receptor can be highly expressed in breast cancer where it's linked to endocrine resistance. We know that the wild type receptor is expressed in pancreatic carcinoma, and in that tumor it's linked with invasion, and particularly perineural invasion. But we also have mutations that happen in this receptor that can be linked to various phenotypes. So rearrangements of the RET gene, which result in a chimeric form that sits in the cytosol, not at the membrane, are found in a subset of lung adenocarcinomas, and also are very frequent in papillary carcinomas. So those are, again, regulated. But what we're going to talk about today are the mutations that happen in the neuroendocrine tumors, and specifically in medullary thyroid carcinoma, where mutations of RET that are point mutations that lead to activation of the receptor are found in a disease syndrome called MEN2, or multiple endocrine neoplasia type 2. So MEN2 is an inherited cancer syndrome. That means that it's passed from generation to generation. At the bottom here, I've shown you a pedigree of a family that we've worked with extensively in the past, which has the MEN2 syndrome. And what you can see here is that the tumors that are primarily associated with this are medullary thyroid carcinoma, shown in black symbols here, and pheochromocytoma with the hatched symbols. And you can see that it's very clear autosomal dominant inheritance pattern from affected parent to affected child. The disease is very early onset in some of these cases, so it can be early in childhood or young adulthood. The disease first manifests itself. And these individuals are often have multifocal or bilateral tumors associated with the disease. What's novel or what's unusual about MEN2 is that the mutations that we see that are giving rise to this disease are mutations in an oncogene, not a tumor suppressor gene. And that has a number of different kinds of implications. So if we break this down a little further and look at MEN2 a little bit further, there are actually two very major subtypes of the disease, and we're going to talk about those distinctions. I'm going to discuss in a little more detail. MEN2A is the relatively less aggressive form of the disease. It's characterized by parathyroid hyperplasia, and then medullary thyroid carcinoma, and an adrenal tumor that's called pheochromocytoma. This disease is relatively less aggressive, although it's still quite an aggressive disease. But the onset is the later of the two. So later onset in this case means in teen years of early adulthood. So you can decide whether you think that's later onset or not. On the other hand, MEN2B is a much more aggressive disease form. It's much less frequent, but is associated both with the thyroid and adrenal tumors and also with other anomalies, including metamorphinoid habitus, a long, elongated body shape, a neuromas of the mouth and the intestine, and other features as well that are associated with this. This is a more aggressive disease, much earlier metastasis. Early onset in this case can be very early in life. In fact, I think the youngest cases that have currently been reported are under 6 months of age. So it's a very, very early disease. Both of these syndromes, both of these phenotypes are caused by mutations in RET receptor. So the pattern of these specific mutations and the disease that they cause are very, very specific. So in the case of the MEN2 cases, more than 85% of these cases are caused by specific mutations of cysteine residue in extracellular cysteine 634. And as a result of those mutations, these receptors are able to dimerize in the absence of their ligand. So they're constitutively activated without having any ligand and can be form dimers on them. In the case of MEN2B syndrome, the mutation is in the intracellular domain, in the kinase domain. And this mutation actually releases autoinhibition of the kinase, so the receptor is always partially activated. And as a consequence, that receptor can be active as a monomer or can be active as a dimer, which is the usual receptor tyrosine kinases. So it's much harder to regulate because it's constitutively active. It's always on. In fact, both of these receptors, although they're quite different mechanisms, lead to constitutive activation of the RET receptor. But they also have, but because they're very different, they do very different things to the receptor, we have to have some other reason than just constitutive activation that's leading to the differences in these phenotypes. And there's a lot of evidence that we have now that suggests that, in fact, it's not just the presence of the mutation that's there, but also the changes in regulation and localization, interactions of these proteins because of the different modes of how they behave, and differences in the ability of these proteins to be down that are all contributing to the relatively different presentation of diseases, different effects of the two different kinds of diseases. So I'm talking about these terms of regulation in terms of localization, interactions, and downregulation. So how are these things different? And in order to maybe talk about that, we're going to have to take a little step back here and talk about how the normal protein functions so we can see how the mutants are different. So the RET receptor, we talked about it a few slides back, is activated at the cell surface, as are most receptor tyrosine kinases. It binds its ligand here at the cell surface, becomes autophosphorylated. And the protein then is often regulated for downregulation and for targeting of its signaling through the process of internalization and trafficking through different cellular components. And that happens, at least initially, through a process called clathrin-mediated endocytosis, or CME, in which these receptors that are on the cell surface act as cargo for recruitment of molecules that form a lattice associated with them. And those are clathrin proteins here. They're recruited by adapters like AP2 and RET, and they are a receptor, and these adapters and clathrin form these pits into the cell surface called clathrin-coated pits, which eventually are able to pinch off the surface of the cell and move into the cell, so our receptor, still active, is here within the cell. These vesicles can then fuse with different compartments of the intracellular lysosomal endosomal pathway here, and they can be targeted to different areas of the cells for different processes. So some of these vesicles, some of these proteins are, when these vesicles are fused with the endosomal compartments, the endosome, proteins can either be recycled back to the cell surface, they disengage from the ligand, they are dephosphorylated, and they can move back to the cell surface where they can then begin this process again. Others of these proteins will then go on through a series of compartments, early and late endosomal lysosomal degradation, and eventual termination of that signal and loss of that protein. So this is a very elegant way that protein amounts on the cell surface, signaling and location of those signalings can be altered and can be regulated within the cell. We've done a lot of work. This is work that I'm going to highlight from Randy Heineman, research associate in my lab, and Doug Richardson and Matt Krupke, previous PhD students in the lab, who really teased apart how the wild-type RET protein actually goes through the pathways. And using some confocal microscopy, in this case, looking at red in red and clathrin in green here, Matt was able to show that, in fact, after we activate our receptor, it only takes about 6 to 8 minutes for RET to begin to be recruited into these clathrin-coated pits in order to be downregulated and internalized into the cell. Once it's activated and is beginning to move into these pits, we can look at how the protein is internalized. And this is an assay here where we've isolated only the protein that has been internalized into the cell, looked over time at how that protein accumulates. And you can see that within 5 minutes here in this Western blot, that we begin to see protein accumulating intracellularly. So It's moved from the cell surface and into these internal compartments within 5 minutes and continues to internalize and accumulate for 30 minutes maximum. So it's rapid process moving into the cell. Once it's moved into the cell, you might think, well, that's the end of it, it's finished signaling. But in fact, a lot of the signals that we see from receptors actually happens not at the cell surface, but intracellular. And so you can see that. This is somewhat from Doug Richardson's work. If we look at cells in which we have expressed red protein, and we look at cells in which we have mutated dynamin, which is this protein that pinches those vesicles off the cell surface, you can see that while we have phosphorylated ERK in cells that are wild in response to the ligand, or if we overexpress dynamin, in the absence of dynamin, we don't have any signal to ERK. So in order for ERK signaling to be achieved downstream of this receptor, we actually have to have that receptor internalized to those internal. So signaling is regulated internally in the cell as well as at the cell surface. Once the cells have reached the endosomes and they can also be recycled back to the cell surface. As I said a moment ago, this is just an example of cellular fluorescence showing a marker for the membrane itself here in green, in red, and you can see that they localize on the membrane 0 to 1. But after 5 minutes, I hope you can see that in fact the red that's on the cell surface has vanished as that red protein has been internalized. And within 20 minutes, you can see that the red is returning back to the cell surface again. So red is being moved into the cell, it's being retargeted for the membrane, and it returns to the membrane within a few minutes of signaling. Okay, but not all the protein obviously goes back to the cell surface, and another major portion of it, in fact, the majority of it, is targeted for degradation. See that it becomes ubiquitinated with these black streaks. Yes, that is what that's supposed to look like. Demonstrating that in fact the majority of it is actually probably being lysosomal. So that represents a sort of snapshot of a number of years of studies on how the wild type red receptor is actually regulated through the different steps of the process of internalization and downregulation. So how does that differ, or does it differ from how the main two proteins are regulated? So this is work that was primarily done by Tim Walker and also by Eduardo Reyes, previous PhD student in the lab, but it also reflects the work of a number of different people as well over the years who've contributed to this. So the first thing that we see that is different in how the MAN2 proteins behave is early in their production. So MAN2 proteins, in particular, in this case, it's the 2B red protein, don't need to be dimerized, don't need to have a ligand to be activated. And we can actually show that these proteins can actually be activated before they reach maturity and before they actually reach the cell surface. So this experiment here is using a series of transient transfections of very short periods, putting the wild type red protein in these cells or the 2B red cells. And you can see that after 2 hours, 4 hours, 6 hours, you can see the beginnings of the expression of the wild type. And you can see the total red protein being made and approximately the same kinds of rates in both of these different constructs. But if we look at the phosphorylation or activation of these proteins, which you can see is the wild type protein, needs to be present for about 8 hours before it will begin to be phosphorylated and begin to activate. So that represents about the time it takes that protein to reach the cell surface. The 2B red protein, in fact, is activated almost as soon as it is needed. So it doesn't need to mature at the cell surface. It's being activated in the Golgi. So we've got actually active receptors in a different part of the cell than we would normally see them. And they're actually activating very much early in the process of maturation of that protein. So we got a much longer timeline. When we look at these cells and actually look at what is happening in these cells in terms of red is being expressed, you can see that in wild type cells, this is done with a surface intensity of red using microscopy. You can see that in fact, surface levels of red in wild type cells, plus or minus 2 GNF, are present, but that in fact there is much, much more 2A red and 2B red surface. So they're maturing to the cell surface and accumulating the cell surface, the wild type, more than the wild type protein. When we look at the process of, although these are actually proteins, they're not being activated by the ligand, but they still can be recruited through this internalization process into early endosomes. And I know that these images aren't going to show up very well, but you can look at the summary down here. So hopefully you can see here that we have the merge of red marker for the endosome, RAD5, and you can see in the wild type very little colocalization, very dramatic colocalization of 2B. You see that quantified down here at the bottom with significantly more internalization of 2A and 2B. So they're moving actively through this process as well. Not everybody. And then if we actually look further at the recycling of these proteins, this is a little dull look at bar graphs, but bear with me for a moment. Remember I showed you that internalization was very little by 5 minutes of treatment with GDNF, but by 30 minutes of GDNF, we see that there's a lot of internalization of these proteins. And then in fact, the 2A protein and the 2B protein internalize much more than does the wild type. But if we look at the proteins that have recycled, and we can do this by looking at the specific compartments in which these proteins lie, we see that in fact, the 2A protein recycles much more readily back to the cell surface. So we have proteins starting earlier, we have them accumulating on the cell surface, and we have them moving into the early endosomes, but then moving much more robustly back to the cell surface. So we're accumulating a lot of that protein on the cell surface in a dramatic kind of way. So what does that mean for these cells? Well, if you'll recall, I mentioned that the wild type red protein was recruited into these domains of lipid raft by recruitment through these glycosylphosphatidylinos called late adapters, co-receptors that are here. And this is where the signaling happens. We don't see the wild type red receptors move into this domain in the absence of ligand. They don't have anything to force them into that domain. And that's what we see here. This is a focal image showing the a marker for the GM1 ganglioside, which is partitioned primarily into lipid rafts and membranes with some modifications, primarily in lipid rafts and membrane. That's in green here and red. And you can see that they don't colocalize. We don't see a lot of wild type red in these membrane domains. In the case of the MEN2A mutant on that, I hope you can clearly see the bright yellow here, where we're seeing the majority of our red on the cell surface is actually localizing these lipid rafts. So these mutants are moving into the dynamically moving into those signaling domains very rapidly without requiring activation or ligand to move them into that position. And we see the MEN2B is also much more highly active in these domains than the wild type, consistent with both of these things being constitutively active and constitutively signaling through those same pathways, stream, or potentially through novel pathways as well. So we got a lot on the membrane. It's in those active domains. You'd think that it might be signaling. What might the impact on moving these proteins into different domains be? And in fact, what we see is that all of that excess protein, all of that recycling actually changes the half-life of these proteins. So the wild type red protein shown here, this is a cyclohexamide time assay. The wild type protein shown here in blue, it's got a relatively short half-life compared to the 2A and 2B, which are much more long. So more protein on the surface, more recycling on the surface, and a much longer half-life on this protein. What does it do in the cell level of cell signaling? So this is a very lovely assay that was done, which is a collaboration between Tim and Eduardo and Randy. Because most of the studies that we've done here have all been done in thyroid cancer cell lines, which have either the MEN2A or the MEN2B mutation. We've tried to put these into the same cell model using a wild type cell line, which was a neuroblastoma cell line. And Brandy was able to knock out that cell line very effectively. And then we did add backs with either the wild type, the 2A or the 2B protein. So we could actually. And what you can see here, I hope, is that we see very much higher signaling from the mutant proteins. So here we have wild type activated by GDNF in this column. As you can see with the 2A and 2B are much more highly phosphorylated, much more active, activated from phosphosarc, phospho-ERK, and the S6N2 pathway is also highly active. We've got an anomaly in here, which is really interesting, and that's something that Tim is pursuing, and I'll let him talk about that the next time he gives the department a summary. So, but it looks like we have a really significant difference in the signaling pathway between mutants and the wild. That may be a portion of why these are so different from each other. Okay, so sum that all up, maybe. We know that these proteins are mutant and that they are constitutively active, but we've also been able to show that the oncogenicity of these N2 ret forms is also affected by when that activation occurs in the life cycle protein, where that ret signal originates, so what compartment is it in the cell when it begins to signal, and how long does that signal last? How long is that signal from the time it initiates until the time the protein is degraded and moves that signal? So all of those things are contributing to the oncogenicity of these MEN2 mutants. So the question arises for us is, could wild type ret do exactly the same things? Could activity mimic the MEN2 ret by altering any or all of these factors of where ret is and how it's being regulated? So in order to ask that question, we're actually going to move from the thyroid cancer to a different tumor, which is also a neuroendocrine, and that is a pheochromocytoma. So pheochromocytoma, or pheo, or PCC, is an adrenal tumor that comes from the chromatin cells of the adrenal. This is an active secreting tumor. It's often, you've probably seen it on whatever medical drama you prefer to watch. Because it's one of those really sexy things that nobody can figure out what it is until suddenly there's some sort of crisis, and then Dr. House or Dr. Sex Dreamy comes in and says, it must be a pheochromocytoma, and everybody cheers. So this is the tumor of choice for that kind of a program. Pheochromocytomas are very frequently hormone secreting. They secrete epinephrine, adrenaline, they secrete norepinephrine, or noradrenaline, they secrete dopamine. And they're associated with hypertensive crisis, with headaches, with hypertension, all of those things that can be associated with many other phenotypes as well. So they can often not be recognized for a reason. One of the really interesting things about pheochromocytomas is it is the most genetically determined tumor that is known. Over 40% of theos have hereditary mutations in one of 25 to date susceptibility genes. And these genes are they have various different kinds of functions within the body. They have variable penetrance and they have variable risks of malignancy associated. So we're not going to talk about all of those. Genes fall into three clusters, really the pseudohypoxic signaling, which involve the succinate dehydrogenase genes, the Wnt signaling pathway, and the one we're going to talk about today, which is the kinase signaling pathway of pheochromocytoma, which includes RAD. The kinase signaling cluster of pheo genes includes, again, a plethora of different kinds of proteins, from enzymes to receptor tyrosine kinases, adapter proteins, transcription factors, and proteins of unknown function. RET, here at the top, is amongst other receptor tyrosine kinases in this cluster, but we're going to talk about another protein that I would hazard a guess most of you have never heard of, which is a protein called TMEM127. So TMEM127 is a tumor suppressor gene in pheochromocytoma. It's also been related to renal cell carcinoma, which we're not going to talk about today. It's a multi-pass integral membrane protein. It's found in the plasma membrane and also in membranes of essentially all of the lysosomal and endosomal compartments as well. What its role is really not clear at all, but it's thought to have some role in endosomal cargo trafficking. When you have mutations, the pheochromocytoma mutations, they tend to cluster within the transmembrane domains of this protein. So they tend to be within the membrane spanning motifs. And as a result of these mutations, we see that the proteins are lost from their membrane compartments and they're found move into the cytosol. And this is just down here showing that. Here's a marker for the early endosomes where TMEM is, TMEM in green. Normally we see these co-localizing here in yellow. But in the presence of these mutations, what you can see is that there are a few speckles of these, but they're not in the endosomes. And then most of the staining for TMM is diffuse and through the cytosol. So they lose their ability to move into the membranes in those positions. So given that REP is regulated, at least in part, by regulating through these endolysosomal pathways, and given that TMEM is a regulator of cargo sorting, maybe, and given that they both cause diffuse cell homocytoma, seemed like a logical thing to ask what the relationship of these proteins might be. And so in collaboration with Patricia Daya at University of Texas, San Antonio, we were able to see some IHC of pheochromocytomas of various genetic origin. So these are tumors with snaky hydrogenase A mutations, RET mutations, and then several T mutations. And what you could probably see immediately is, although there's lots of RET in the RET mutant positive tumor, there's a lot more RET in the TMEM positive tumors. Okay? So TMEM negative, I guess, because these are TMEM knockout tumors, right, where they've lost the tumor suppressor. So we're seeing that there's more RET in tumors that have no TMEM receptor. So in order to have a look at this, Patricia generated a cell line, which was the neuroblastoma wild-type cell line that I just showed you for a while back. And she generated a CRISPR TMEM127 knockout cell. And what you can see, this is work from Tim, is that in fact there's a dramatic increase in TMEM protein, RET protein in these TMEM knockout cells. This is at the protein level, not at the transcript level, because in fact, if anything, there's less of the RET transcript in these cells. So in the absence of TMEM, we see a huge increase in the RET protein in these cells. We could also see where this protein was localized. So this was an assay I showed you before, where we can actually isolate just the proteins that are on the cell surface. We do this by tagging all the proteins on the cell surface with biotin moiety, and pull the biotin out, and that means that protein was on the cell surface and not anywhere else in the cell. And what you can see clearly here is that in the knockout cells, all of the RET is on the cell surface. So there's actually a 5-fold increase in the amount of RET on the cell surface if you don't have TMEM in the cell. So what we're seeing is very reminiscent of what we saw with MIN2B. We've got a normal RET protein, but it's accumulating on the cell surface to an extensive degree. And just like we saw with MIN2B, our protein that's accumulating on the cell surface is a much longer half-life than does the wild. Well, they're all wild protein, but then does the protein in the cell where TMEM is right. Okay, so RET's accumulating on the surface. It's got an increased half-life. Why is the RET protein aberrantly localized? What is happening to it to keep it on the cell surface? So in order to look at that in a little more detail, called on a collaborator from Metropolitan University, Kostin Hopnesky. And Tim went to Toronto and spent the whole summer in Toronto working with Kostin using his turf microscopy to look at what was happening to RET. So he was looking specifically in this region of the cell. So Tim used turf microscopy to look at this. That may not be a term that's familiar to everybody. Turf is total internal reflection fluorescence microscopy. And this takes advantage of the fact that you can use, instead of passing light directly through a sample so that you can look at the whole depth of the sample, you can use laser light and bounce it off the critical angle of reflection off of the surface of the cell of the cell that is sitting in the microscope. The light will bounce off of that, but it will cause electromagnetic radiation will cause any fluorophores just at that interface to become fluorescent. But it won't touch anything that's further into the tissue or further into the section. So you can look at a very good snapshot just of what's in situ on the cell surface or in these tissues. And so what Tim did was he looked at clathrin, which is EGFP clathrin in these cells, red clathrin and RET stained on the cell surface. And what you can see is non-knockout cells. We have a lot of, when we have no GDNF, we have some localization. We add GDNF, clathrin and RET come together because they're trying to do this to the cell. When we looked at TMEM knockout cells, We don't see anything. You can see this quantified down here. Red and clathrin on the cell surface. You have GDNF that goes up as the red moves into the clathrin pits. GDNF in the TMM knockout cells, we don't see polarization. So red's not moving into these clathrin pits. And if you actually look at live cells and look at clathrin movement on those cells, you can actually see that in the wild type cells, we can have all of these little pump as clathrin comes together to make these little pumps, and then moves into the cell and disappears from the surface of the TERT image. So it moves out of sight of the microscope, the TERT illumination. And the TMM cells, you can see that do not have many of them, they go around, and they don't move into the cell. So we're not getting clathrin-mediated cytosis happening. These proteins are not moving into the cell. So clathrin-coated pits are pretty small. You can look at them with these kinds of puncta, but it's pretty hard to look at individual clathrin-coated pits. And so in order to do that, we collaborated with Doug Richardson, who's at Harvard University, and who's a longtime Ret fan. And he did some structured illumination microscopy for us. So this is a form of super resolution microscopy that allows you to look at bodies, structures that are in the 60 to 100 nanometer range. And he was able to look at again, the same kind of TERT image. So just at the cell surface, if you look at the presence of clathrin, it's looking at the cells that were generated by Brandy in the lab, expressing clathrin enrichment. And what you can see here, if you compare these two, I like to think of these as jelly beans. So what you can see in the TERT knockout cells is that there are many fewer jelly beans and that they are much smaller jelly beans. Whereas in the knockout cells, we see a lot more of these clathrin-coated pits, and they're much larger. So the normal cells, the knockout cells here, are really happy making these clathrin-coated pits. The TMM knockout cells are just not making these vesicles to internalize and bring proteins into the cell. As quantified down here, you can see small, the size of the CCPs is very small in the knockouts, and the numbers are very small. So we have a defect up here somewhere in terms of internalization. These proteins are not moving into the cell. So for those of you who are aficionados of cell surface and trafficking in cell, you're probably saying, okay, but lots of things internalize into clathrin-coated pits, and lots of things move into the cells through endosomal compartments. How do you know this has anything to do with Ret? And that's a really good question. And so that was the next question that Tim asked. He asked, is Ret the only protein that moves this way? And in fact, if you actually use the TERT images to look at Ret on the cell surface, you saw that there's lots of Ret on the cell surface of wild type cells with red GDNF. It goes into clathrin pits and moves inside the cell, so it decreases. But the Ret intensity in the cell surface in the human knockout cells is high and stays high. In these same cells, you can actually ask the same question about EGFR. There's a lot less EGFR in those cells, but it does the same thing. It stays on the cell surface in the knockout cells. There's a lot more in knockout cells. So there's multiple proteins that this is done with. And in fact, then Tim and Brandy went to the freezer and they pulled out every antibody that we had to hand that would recognize a cell surface protein. And in fact, what they saw was a really uniform phenotype. So if we look just at the cell surface protein, we can see that there's an accumulation of Ret, of EGFR, of cell adhesion molecules like interferon, beta-3 integrin, and even proteins like the transferrin receptor, and they're all accumulating in the cell surface because they all normally internalize into the cell through clathrin-mediated endocytosis, and we've screwed that up. And we haven't teamed. Okay, so we have a defect that's preventing all of these proteins from moving around normally into the cell and on the cell surface. So what exactly is the defect? How is this being defectively forming these clathrin-mediated endocytosis? So we, again in collaboration with Mustin Antonescu and his postdoctoral fellow, Michael Sugiyama, we actually try to interrogate this at the single molecule level. So Michael was able to use fluorescently tagged FAB to the EGFR and use it to track single molecule movement on the cell surface using TERT microscopy. So this is just an example of what that might look like on the side here. On the upper panel is the movement, and these are individual tracks. They're tiny little squiggles of individual EGFR molecules moving on the surface of this cell. In the case of the TMEM knockout cells, what you can see is all of these tracks are much longer. So we've gone from single confetti to streamers in here. These are all much longer tracks of these proteins because these proteins are moving much further, so they're more motile, diffusing more effectively. And that's just quantified here, where proteins that are constrained and can't move don't change in their diffusion in our TMEM knockout cells. But if the proteins are confined but not immobile, or if they're freely moved, they diffuse significantly. So they're zipping around a whole lot more than the subsurface of the first surface of the cell. You don't have to move them. So how does that? Let's go back to clathrin-mediated cytosis and see if we can figure out how that. So CCP formation and internalization is a multi-step process. It doesn't just happen like that. Clathrin proteins, the clathrin triskeleons here, the little green ones here, and the adapter proteins that they're associated with are near the membrane and back the membrane all the time. And when they begin to come together in terms of trying to nucleate into clathrin-coated pits, they form these little stochastic assemblies. Some of those will be successful and recruit more clathrin and more adapter protein to actually begin to form the initiated clathrin-coated pit. And some of them will fail to recruit enough carbon and they'll disintegrate and redistribute, so we won't see any formation. Even these initiated clathrin-coated pits don't all form into maturity, but they need to recruit enough carbon and enough clathrin to make a productive pit that will internalize and eventually reconstruct the cells. So there's lots of steps at which this could fail. And what we see in the TMEM knockout cells is that these stochastic early assemblies, these little tiny accumulations of clathrin, are much more frequent in these cells, which might fit with the fact that the cells seem to be more motile, the membranes seem to be more motile, and these proteins could be moving around more quickly. But they fall apart, and we don't see nucleation and initiation of clathrin pits significantly efficiently. So these are much less able to form these clathrin pits. But if they do manage to recruit enough clathrin to make these pumped out, to make these accumulations of clathrin, then they tend to be more stable. And so they go on to make very inefficiently. They tend to be to mature more effectively than do the wild cells. Then they do the wild cells. But so more efficiently, but less frequently. Okay, so we're looking at what looks to be a defect of the ability to form clathrin-coated pits because proteins are moving too quickly to be captured as cargo and taken into the cells through clathrin-mediated endocytosis. So how does that impact the cell? Well, in these cells where TMEM is knocked out, what we see is we have, again, this excess of RET protein, and we see an excess and increase of RET phosphorylation. We also see that it's constitutive, so even without any kind of ligand at all, we see constitutive activation of that receptor. And we also see constitutive phosphorylation of AKT, and ERK, and mTOR activation of M6 is up there. And all of this in these cells is dramatically upregulated because RET is upregulated and captured on the. And we know that this is RET dependent, even though there are many other receptors and other signaling pathways on the cell surface, because if you add a RET inhibitor, you knock that out. And Tim used 3 different inhibitors to show that this was very clearly RET specific. He used vandetanib, which is a multikinase inhibitor, and then 2 selective RET inhibitors, completely out of RET phosphorylation and phosphorylation through these pathways. So we have constitutive signaling. It's downstream of RET, even though RET is not mutated in these cell types. And so how does that fall out? What happens next? What you can see is that RET is actually driving an MN2-like phenotype in these cells. And in fact, if you look at proliferation of these cells, our knockout cells don't proliferate very quickly in the absence of GDNF, but if you add GDNF, they proliferate more effectively. And the TMEM knockout cells proliferate irrespective of GDNF very significantly. But if you add sulfacatinib, the RET inhibitor, to these cells, what you can see here is both our MOC knockout cells and our TMEM knockout cells completely obliterate the ability to proliferate. So we can kill the proliferation of these cells which have wild type red and not mutant by treating with the red inhibitor. So we can actually use that to limit the proliferation of these cells. And in studies that are quite recent from our collaborator, Patricia Dahlia in San Antonio, she actually took these same cell lines and put them into some mice and showed that in fact she could see the same thing in people. So that you see that the knockout cells actually do grow better, and that when you add some catnip to those cells, you actually can drop back that proliferation of the cells in terms of tumor volume and also in terms of tumor weight. So in fact, this works both in vitro and in vivo to limit the growth of TMEM mutant tumors. Summary, you'd be glad to know. So just to summarize what I told you about, I told you a little bit about the wild type rat and how wild type rat gets regulated, how it appears on the cell surface and it's internalized through clathrin-mediated endocytosis, going through different steps to signal internally to lysosomal deprivation. And then I showed you how MEN2 that is actually different than that. It actually is able to be active before it reaches the membrane. You see signaling from there and accumulates on the cell surface and also in internal compartments, activating signaling through all of those different positions to decrease relatively the level of degradation. I also showed you how TMEM127 can mimic this effect in the MEN2 mutation by accumulating the wild type protein in the cell surface because of the failures of clathrin-mediated cytosis, and so that it can return through these compartments and regulate it. As a consequence, we see that increased activity in signaling and an increased cell penetration. But the good news is we also have the ability here to use that red inhibitor in another population that's never been considered. And we use that to target TMEM127 tumors potentially in the future. Okay, summary. So I just want to acknowledge all of the people that were involved in this work. Big shout out to Tim, particularly Brandy in the room. You really did the majority of the work on the TMEM paper and on the MEN2 work as well, which is ongoing. Eduardo Reza-Veras, who has just graduated, and really did a lot of the work in optimizing the models that we use for the MEN2 work and going forward with that, how that's going to go forward. Other members of our lab, past members of the lab, our collaborators, Matt Krupe, who did most of that work on the early wild-type receptor and is now collaborating with us from Ottawa, Costa Antonesco and Michael Sugiyama at TMU, Doug Richardson at Harvard, and Patricia Dahlia at UT San Antonio, and all of the funding agencies who have helped us with all of this. But finally, as Peter alluded to, I want to acknowledge all of the people from the lab who've been involved in these projects over the years. Those of us who are doing the work today stand on many, many shoulders of people who've come before us in the lab and have worked on developing the models and defining the parameters and trying to establish how these different molecules may work together or against each other to cause the diseases that are associated with that. So I want to acknowledge all of these shoulders that held us high and lifted us up to do the work that we do. So thank you very much.
Oh, thanks. So that was a tour de force, and I just got to say one thing about this. It's nice to see somebody that goes and finds a gene that is mutated in cancer, but then goes back and figures out how the mutation affects function. And I think what we've seen today is a magnificent demonstration of figuring out how things work and why they don't work when they're mutated in specific ways. Yeah. So thank you very much for that. Are there any questions? We do have time to ask questions down in room 101, but maybe one or two. Paul, you had your hand up?
I have two questions. One, I guess the obvious one is if TMEM is ineffective and therefore all activating proteins on the surface are equally effective, is it associated with tumors that are caused by other oncogenes that are sitting on it?
That is such a good question. So There are mutations in TM127 in renal cell carcinoma. The red is not expressed in those tumors, but MET is. So there are other receptors that are likely overexpressed on the cell surface of other tumors that would be drivers in those diseases that are not red. So I think red is a not quite innocent bystander. I think of it as a backseat driver in this case, but it happens to be that in this specific tumor, it's red that is highly expressed. So as a consequence, it drives in these cancers. But in other tumors where TM is mutated, I think it's very likely that you'll see that it's actually in the tumor cells.
And the other question that I had is, I assumed that all the proteins that were in these extensive pathways that are activated like fibroblast are actually assembled at close to the cell surface, but you implied that they're actually probably assembled in the Golgi.
That's where they get made. So they
yeah,
are made.
But you made the point that mutated red is actually activated in the Golgi and implied that that's where it started its activating activity. And therefore, I guess you believe that all of the proteins in the pathway are actually there in the Golgi while it's being activated.
So
do you know that?
So it is activated in the Golgi. And we know that it interacts with several things in the Golgi. It doesn't necessarily activate the same pathways because those proteins may not be associated with that compartment to allow it to activate those. Okay. So just because it is active there doesn't mean it's making the same signals as it might from elsewhere in the cell, like from memory or from the endosomes.
So what you're saying is its activation of Golgi may have nothing to do with its increased activation time that causes the tumor.
And maybe actually a totally different pathway that is activated. Yeah.
Well, thanks. Normally when we're talking about activated oncogenic protein, we're talking about somatic mutation. The cancer has a mutant protein, but not the feeding cause of tissues. You made the point that most familial cancer suppressor mutations. So this is kind of a unique situation. I'm curious about phenotypes either at the cellular level or organism level associated with just expression of activated oncogenic protein during, say, genesis or physiology, etc. So you just comment on phenotypes associated with pre-neoplastic state of disease.
So we know that when red is activated by ways of mutations, we often see hyperplasia of some of the cell types that it's expressed in. So the C-cell hyperplasia fibrin is very frequently a precursor to thyroid carcinoma. You'll see that very early on in these individuals. It's not at all surprising to see in the two mutation that will have problems with nerves in the gut and also in the buccal patterns on the tongue and the lips. So those are not neoplastic, but they're overgrown associated. We also know that there is different sensitivities in tissue types to the levels of that expression. So we see that typically in terms of cancer mutations versus mutations that cause the inactivation of red related to Hirschman's disease, where the protein is inactivated, or in genital abnormalities tract, which is also a developmental effect. It can be associated with red function. And so it's very, very clearly that more red protein in some tissues doesn't affect those tissues, but less red protein in those tissues. There's also a really elegant kind of story associated with that mutations that we see too. So some of the mutations, I talked about two that are most common, but there are rarer mutations, they're white mutations that affect, for example, upper cysteine residues in the extracellular. And they are transforming, but a much lower portion of that is they don't fully. So although the protein is made and it's constitutively active, it doesn't effectively go to the cell surface as efficiently. So there are some of those mutations that actually cause both Hirschman's disease, which is a phenotype of lack of protein, and thyroid cancer, which is a phenotype of overexpression. So you can see with the way these things can titrate that It's tissue specific, very specific, cell specific things type associated.
Back to Paul's question. That was the 2B form that's activated. So it's autophosphorylating. Does that block ubiquitination? And is that what's just causing an upregulation of the whole signaling pathway? So it's presumably a degradation of that ubiquitination.
Ubiquitination would be related to degradation. But ubiquitination usually happens so
If you had a fork that comes into the membrane, either getting recycled back or going off towards degradation.
So you're asking me, whether it's an action.
Sure.
And we did see that the 2 protein was recycled. So.
Very excited to see the familiar faces. Yeah, it's very exciting.
Make you feel old that you recognize most of them.
Very exciting. I just, when you were talking, I was thinking about it. Membrane protein, right? They can function on the membrane protein, signaling function. But when they pack it into the cellular, they have a different function. And they can also pack it through the nucleus. So what's happening to that?
Does red go to the nucleus?
Yeah, how they're rapidly.
So it's a great question.
How they function?
We looked extensively and could not find any evidence that that was in the nucleus. We did a number of different mutations. We looked at a number of different antibodies to see if we would identify any mutation. Yeah. There was a significant level of red in the nucleus. We weren't able to verify that they were red. So it could be that it's a portion of red for which we don't have an epitope, we don't have an antibody that recognizes. So it could be that something like that happens, but there isn't any data that we could generate that suggests that that's. Yeah. Anna.
Yeah. Yes, I really was fascinating to see this connection between mutation, single mutation pathways, and actually cells. Even looking at the images, that was amazing. I wonder if image processing can actually detect mutations.
I don't know. You have to do
but
that.
Anyway, this is not my question.
Okay.
So, yeah, my question is about mutations. So first, I would say question that you said 40% of the second type of cancer, they are inherited mutations. Whether you see the difference between inherited and somatic mutations in terms of what they actually, where they occur and what they do. If you have any overlap, you see any overlap. And actually, in general, I wonder why this particular cancer type is 40% of inherited component. And my second question is about this, whether you see or anybody actually investigate the occurrence of mutual exclusivity red mutations and this TMEM mutation.
So 2 good questions. Second one first, because it's easier. No one has seen a co-occurrence of the 2 mutations, of a red mutation and a TMEM mutation, but that's not too surprising. TMEM mutations are found in about 1% of pheochromocytomas, and red mutations are found in about 8% of them. So the probability of having both in one tumor is not likely. We did not try putting the combination together. I think that is being really important to try. So I think it's probably unlikely that you would find both, but I would predict if you did find both, it would probably just as you have a further mechanism of increasing the subsurface rather than strong. First question in terms of pheochromocytoma and 40% hereditary mutations. So there's 3 clusters of types of mutations of the pseudohypoxia pathway. and in these kinase clusters, you see varying frequencies of mutations in those three that vary different phenotypes associated with them. You do see overlap of the sporadic mutations, and so you see them in the same genes. They are, I believe, all there are RAS mutations and MEK mutations which can be activated. All of the others are inactivated mutations. So they may not be identical mutations found in sporadic and familial, but they're functional. There's no protein. So yes.
Maybe I'll ask the last question and let's see other hands. So you did an elegant job of showing how dysfunctional RET contributes to cancers and other diseases. You didn't get around to the subject that I know you're very passionate about these days, which is perineural invasion. So in this case, it's kind of analogous to how tumors can provoke the ingrowth of blood vessels. So do you look at parallels between that and the signaling pathways that the tumors are engaging in between vascular infiltration or tumor neovascularization and innervation?
Maybe, but I think maybe the way to look at this is probably some of the work that Larissa Oliveira and Julie Shields are doing in the lab, where they're really interested in how the membrane is changed in terms of generating cell outgrowth, cell extension, and invasive podia. And I think that there's more that we can find out when we understand a little bit more about where these proteins are in the membrane and how they're functionally aberrantly working in the membrane in terms of these cancers that may also be relevant in how they're driving other processes, specifically engagement with matrix, release of matrix metalloproteases, and formation of invasive structures, all of which can be argued and may be relevant to how proteins are repositioned in the membrane, what membrane domains they're in, and how they signal specifically from these different domains. So I think there's overlap considerably between these, even though one is causation and one is.
But do any of these tumors that invoke this invasion, neural invasion, do they produce GDNFs? Do they? These
ones, no.
No.
These ones do not. The invasive ones, when we're looking at nerve invasion, those ones we actually have a lot of autocrine activation. So we have tumors that are expressing both the ligands and the receptors and can actually be self-perpetuating and driving that process. Be afraid.
Paul,
a
personal question. There's a family that I think whose tumors you worked on that was in Kingston that I knew when I first came here in the 70s, who had the MEN2B, and we used to see a lot of medullary carcinomas. And did that family have any influence on you choosing this as your focus, RET?
So I guess in a sense, so for the students here, when I came here as a student, there was a faculty member in pediatrics, who was one of the first women to be a graduate of biochemistry at the University of Toronto. And she was, I believe, the first woman in research here in health sciences. She was quite, and still is, a quite exceptional woman. She had a passion for medical and ways of those families. She did a lot of legwork, going out and collecting samples from those patients, working with them to identify potential ways to break that causes of that disease and how the gene therapeutics might be developed for them. Because we talk about it not in a trivial way, but in a sort of casual way. And two is a very, very awful diagnosis to have now. At the time when I began working on this, the gene wasn't known. So if you had a family with 3 children, 50% of them would have the disease, and you as a parent knew that half of your children would have the disease, but you had no way of knowing which ones might develop them too. And there wasn't a good diagnosis test, and there wasn't a good predictive test. The test that was used was a stimulation assay. And so your kids would come in You can move your kids every 6 months, and you will do a stimulation test to see if you can get an increase in hormone secretion in the body. And if you had an increase, that meant you had a tumor. So you weren't actually looking to prevent, you weren't actually looking to identify before disease, you were looking for the start of the disease. And it's easy to imagine how you would bring in the test. Wasn't pleasant, it wasn't super awful, but it wasn't pleasant. Kids didn't like it. You can imagine that kids every 6 months for that. For a while, you do it, and then kids would pick, and you miss one. You may be I miss one, you get this too. And the next time, you have a positive test. The child needs to have their thyroid. And these diseases are slow growing. They're not fast, but they metastasize very early. So these kids would miss one of these tests, and then the next time they might have metastatic disease with the other nuts, with metastases down through the organs through the breast. So then it was really significant life changing. So identifying a gene for this and being able to test and say, at least you aren't going to have it at all, and you can stop having this test. Or you are going to have it. We'll monitor and figure out a way to do that diagnosis for you, that tumor removal. That was life changing. So personal story here. This past year was the 30th anniversary of the exchange. And I went to give a talk in Scotland in Glasgow last November and on this subject. And when I came to the end of it, as I was going about, I had 2 endocrinologists come up to me and say, you have no idea what you've done for my patients. That my patients, you changed their lives. Thank you. I think it is something to be aware of. This seems esoteric. It's in the lab. It's Western blocks. But to a patient, making the difference between a diagnosis before there is tumor everywhere, the diagnosis that could prevent it, because we can treat the thyroid hormone replacement. You can live with that very comfortably. So
I'm telling you, you're never going to get to read the
They're never going to get it. Anyway, thank you.